So as I said, we are going to move forward with the speakers, and after we are going to have the panel. So please take a note of your questions for later. Inside your bags you have notebooks and pen, so you can use it. So let's go to the next speaker. I would like to present here Professor Dr. Christof Scheid. I hope I said correctly. He's head from cell therapy at University of Cologne in Germany, head of stem cell transplantation and the myeloma program since 2003, and qualified person for autologous stem cells and leukapheresis products. Now let me welcome Professor Christof with the presentation entitled CAR-T Therapy in Practice, Clinical and Manufacturing Challenges. Thank you.
Yeah, thank you so much for this kind invitation. It's actually my second travel to a live meeting. Otherwise, it's all online, so it's quite exciting, and I think it's good to meet in person again. And I'm grateful to come back to the UK. Actually, I spent 2.5 years in Manchester at the Patterson Institute of Cancer Research. So, although being German, I still have a kind of emotional link to this country, and I love to see things coming to fruition. And I was very inspired by the Catapult project, because I recognize many of the problems that we face in implementing advanced therapies in the clinic. I want to really go to the clinical side. I'm treating patients with cell therapy, and so this is the ultimate goal of any kind of complex infrastructure to bring therapeutics into patients. So myeloma is a deadly disease, really. In the 90s, patients lived about 2 years in median with this disease, and they were kind of dying in front of our eyes, getting sicker and sicker, because this disease occupies the bone marrow. You see these roundish cells there, and this destroys the bones. Bones break, and the kidney function goes down, and hematopoiesis goes down. So it's really a very dangerous disease and slowly killing patients. Now we are lucky to have a lot of drugs available. Chemotherapy doesn't work much in this disease. And so in first-line therapy, patients can expect to have disease control for about 5 years. So it's more like a chronic disease now these days. And we can remix the combinations. We have 3 and 4 drug regimens we can mix. So we keep being creative and repeat treatments. However, after multiple rounds, patients get more and more resistant disease. And so in younger patients, so after around 10 years, they come to my office and say, listen, I'm fit, I'm willing to have therapy, what do you have for me? And then I have to think about these kind of figures. This is the overall survival of patients after failing 3, 4, 5 classes of drugs. And it's a bit of small print, but you see the survival goes down from 11 months to less than 6 months. So young patients after 10 years of therapy, who's fit, who's in the middle of life, will then have a life expectancy maybe of 5 or 6 months. So that's the starting point where we come in with new cellular therapy approaches. And obviously it's important to have new targets. When you have exhausted all your action points in the tumor cell, it's important to have new targets, and the B-cell maturation antigen is really the prime target at the moment, where many products are trying to attack this antigen because it's quite specific to the tumor cell, and only mature B cells are also in the focus, but we can live without B cells, so it's not so critical to spare these cells. And we have the first approved cellular therapy product. I used to think then this is also valid for UK, but now we have to change that mindset. But at least on the continent, it's approved by the EMA, and so the approval was based on this pivotal study. So we did the apheresis of the patients. Cells go then into general labs in the US. So a lot of freezing and shipping and storage of these cells. Then patients get 3 days of a mild chemotherapy to kind of lymphodeplete or reduce the activity of lymphocytes, and then get the infusion, and after 4 weeks you measure the first response. So it's a very quickly acting intervention. And as I said, these are patients with in a median of 6 years after diagnosis. median of 6 prior lines of therapy, refractory to 3-4 drugs. So it's this kind of hard to treat cohort of patients. And this is what you get with increasing cell numbers. And as you mentioned, you cannot define the dose because you get some back with apheresed cells and they grow or they don't grow. So you can just number these cells in the final product, but you cannot determine this in advance. So this is just what happened in these real patients. And with a higher number, you see you have response rates of more than 80% with the higher cell numbers and complete remissions in about a third of the patients. So these are very exciting response figures that are totally unprecedented in this heavy-to-treat population. And this, I think, explains our excitement to get involved with these highly complex agents. A thing to work on is the durability of response. You see on the left the progression-free survival with the highest dose, it's the upper curve. It goes down, so patients have a response and lose the response. And this is about in a half of the patients happens after a year. So at the moment it's a transient success, I would say, and we have to learn how to improve the success rate and to make this more durable. But if you see on the right the progression-free survival after response, and the upper curve is those third of the patient who had a complete response, you see they are maintaining this response a bit better, but again the curve drops down. So it's not really a cure for these patients, but it's at the moment a transient effect, which is very, very efficient, but there's still a lot of room for improvement. So I could say it's a wonderful success story. We have a new agent, it's very effective, and no problems, but this is just a short list of problems that came to my mind. So it's 350,000 euros per shot. So you can buy a house for that amount of money, yeah. And I just have these discussions. This morning I was emailing with the insurance company from one patient to please say, can you please send the permission to do this treatment and secure reimbursement for that, because no hospital can afford even a single patient not being paid. And if we do 10 treatments, it's 3.5 million euros on our budget as a minus. So we're very aware of securing the reimbursement. Then logistics are a nightmare. You know, we have couriers, we have dates to organize, pick up dates. Couriers don't find the right lab, where in the building is the lab, where I should pick up the cells and these kind of things. We have audits by the companies, we have audits by the payers to check if we are able to provide a high quality of care to the patients. So it's really a demanding task to organize all these logistics. And then timing is a very big issue. So I have to book a slot for the patient. I cannot just make the apheresis and say, here's my bag, can you please collect it? So I need to call the company. Do you have a slot for me? Is it this Friday or next Monday or whatever? So usually it's 2 to 4 weeks in advance I have to book this slot. Then I do the apheresis, so I have the cells in the bag. But then it needs to go to the manufacturing center. I just had an example where it took 4 weeks from the apheresis to the start of the manufacturing process. So another 4 weeks added in delay. I was really mad about this, but what can you do? Then the manufacturing takes 3 to 4 weeks. Then we had this wonderful case where there was Thanksgiving in the US. So I was trying to call the qualified person. Can you release the product? Is everything fine? Can I organize the treatment? But nobody was there. You know, you had nice people on the phone from the call center, but no qualified person. I think they were fishing or so, but these things happen. They say afterwards, they say, oh, sorry guys, it was Thanksgiving, we were taking some day off. So you wait for this release, and this release can take 2 to 3 weeks. If cells come back to the European continent, again, you have a European release. They're not allowed to make the release without having the product on site, so they have to wait for the shipping. Then they do the release, and then I can get the product. So you easily can calculate it can add up to 2 or 3 months waiting time from the thought I want to give you CAR T cells to the treatment with CAR T cells. And this is a very long time in a disease that can kill patients within 6 months. So for me, it's very critical to look at these timing issues. And then really it's a clinical question. Is it justified to make a patient wait for this treatment while being at risk of dying from this disease? And so for me, it's very important, and I really love to see all these discussions about logistics and production optimization to make really these cells more available in a clinical setting, and we need them quickly. So how can we improve this concept? So obviously we could maybe have more efficient CAR T cells, so they have more longer duration of response, they have a high response rate. We could go to rapid manufacturing, and we had the experience with the company Milteny, just 20 miles away from Cologne. They make also trial CAR T cells, and they never freeze these cells. We ship them over by car, just half an hour drive. They are put on the machine, on the Prodigy the next day, and after 14 days I have the cells back and infuse them straight to the patient. So 14 days is a different story than 3 months waiting time. So this is certainly one option to avoid any freezing step and any long distance shipping step. I'm very interested in doing local manufacturing. So I'm trying to have a very small clean room, 80 square meters, a very small local lab, but to be able to take the cells from the apheresis on the same day, put this on the machine, release the cells, and give it back to the patient. It's a year-long process. I know exactly what you mean by staff demands and everything, and you're talking about ridiculous details sometimes with the authorities, but you have to go through that to be able to manufacture on site. I think this is a very interesting model also for the industry to say, why not having a product approved in the EU and then organize local manufacturing as a partnership, because we have this partnership now with the apheresis, so we are part of the manufacturing process anyway, and so for me it's very logical to include also the manufacturing process in a closed system with defined processes. So I think this is a very interesting model for the future. But then obviously a lot of discussion is going about off-the-shelf cell therapy. This is a magic word, and I will show you a few examples. But also I want to mention that we have bispecific antibodies. So many people say, why bother working with cells? Just give the bispecific antibody into the patient and make cells work inside the patient without ever taking them out. So we have to be aware when we develop cell therapies that there's tough competition with very simple molecules, if you will. So just showing examples. So this Silter cell was actually developed by Legend Biotech, and we were first a bit skeptical about the excellent results we got from Chinese trials, but then the US trial, CAR-T1, showed that this CAR-T cell is extremely effective. It has actually 2 binding sites, so I think the binding affinity might play a role in the effectivity of these cells. But there may many other subtle differences might play a role as well. And again, patients were heavily pretreated, and now you're up to almost 100% response rate, and not 30% complete remissions, but 80% complete remissions. So this cell product is extremely effective, and we are very excited to see this hopefully come to the market in the next year. And also progression-free survival. Here you see the 18-month bar, and you see around 70%. So it looks like this more effective therapy also lasts longer. I'm hoping to treat the first patient with this product in the next weeks. So I'm very excited about this. I think it's a very interesting product and maybe more effective than the existing one. And it's going to be very interesting to see, will we ever see a comparative trial? If you imagine the drug cost, that's enormous, plus the trial costs and the risks involved for big companies, it will be very, very difficult to imagine that we ever see a comparative trial of 2 CAR T cells against each other. So it's tricky also from the evidence generation. How about allogeneic off-the-shelf CARs? So one example is today natural killer cells. So you have no T cell receptor, you don't need to be afraid of graft versus host disease. So it could be an interesting way to generate cells, introducing a CAR receptor into these cells. Number of cell sources are possible. You can also do an apheresis of a donor, but you can also use cord blood cells. and one example is developed in Houston at the MD Anderson Cancer Center. They use cord blood cells to generate NK cells, introduce a vector for a CAR against C19, so a classical lymphoma approach. They added IL-15 to make these cells expand better, and they also induce the safety switch. So again, you see the models get more complex. You have not one intervention, you have 3 interventions to modify the cell. Again, you can end up maybe with some safety concerns as well if you modify the cells in that extent. However, the first 11 patients have been treated, and they saw responses in 3/4 of the patients. So the principle has been proven effective in patients, and so it seems to be doable to work with these cells. I know, I have to admit, it's a complex manufacturing, and it's also always a question how good will these cells be, and at what price can you market them in the end? If I can get these cells for 50,000 euros as a bridging option also, then I'm fine. If it's 500,000 euros, then they have to be much better than CAR T cells, and they will not be exempt from running a clinical trial testing against CAR T cells. So the costs are enormous to develop these cells to a clinical level. But again, it's doable. It's interesting. The authors claim we have much less problems with side effects like cytokine storm. But then I would argue, sorry guys, we learned how to handle CAR T-cell toxicity, so for us it's no problem anymore. It was in the beginning. So now with these cells, you solve a problem that we have not anymore. So you see how difficult the changing goalposts are for this kind of drug development. There's another concept to use allogeneic CARs, and these are published data from last year in an adult and pediatric trial in relapsed acute lymphatic leukemia. Again, if you use allogeneic T cells, you have to make a number of manipulations. So you introduce your CAR T cell receptor, they introduce another CD20 mimicking receptor, and they deleted the T cell receptor to avoid graft-versus-host disease, and also they deleted CD52, so they could use Campath as an antibody to deplete the other lymphocytes. So the patient gets Campath and the normal lymphocytes are depleted, and these cells are spared because they don't carry CD52 anymore. So really a complex genetic manipulation. The advantage is you have these cells ready, available, you have them on the shelf and you can use them in patients. And then you can put this into patients, and green you see the response curves. The patient needed Campath as a pre-treatment, otherwise the cells didn't expand. But if you see the little red spots there, these are patients getting an allotransplant. So as a transplant, I would say, great, you have a very effective debulking of the patient, you reduce the tumor burden, and then you do the allotransplant to have a long-term remission. From the reimbursement perspective, I would say, wait a minute, the transplant is at, let's say, 50 or 70,000 euros. So how much can the induction therapy before transplant cost? No way it can cost 500,000 euros. I think from the reimbursement perspective, you'd say, okay, it should not cost more than the transplant itself, if that's a long-term treatment. So we allow you maybe 50,000 euros as cost for this kind of bridging therapy. So when you plan, drug development, you have to be aware, if this is only a bridging before transplant, my price tag must be from the beginning be at a different scale than if I try to develop a curative regimen. So you see how drug development here is closely linked to the kind of application that is planned. And finally, a warning, a signal, I would say. This was another allogeneic CAR trial. In this case, it was transplanted patients relapsing, and then they took donor lymphocytes from the donor of the stem cells and made CAR T cells from these donor cells. So it's a matched setting, and it worked. They got responses, but 2 of the 10 patients developed a lymphoma originating from the CAR T cells. So it's an ongoing discussion. Can we induce by genetic manipulation a cancer cell in the end? And here it's unclear how this happened. But for me, it's a warning signal. We can keep discussing that lentiviral transfer is old-fashioned and conservative, but at least it seems to be pretty safe. And if you use a non-viral system, which is obviously much more cost-effective as well, you might have different problems. And so for me, it was quite an important message. We have to really think not about only efficiency in the lab, things can happen, and here obviously you can speculate. If you co-culture patient cells and donor cells and they match on the HLA locus, it can cause other problems, or it can cause immune evasion maybe. So in the patient these cells are not recognized. So plenty of things to think about, and it just, for me, is a warning that complexity is always also a certain risk for unexpected safety signals. So just as an outlook, I mentioned the bispecific antibodies, bringing together the cancer cell and the effector cell. And in the field of myeloma, it's not as high the response rates as in CAR T cells, but this is an ongoing therapy. You can do subcutaneous injections once a week, once every 3 weeks, and so on. And you see we have 65% response, 40% complete remission. So it's not that far away from the CAR T cell effectiveness data. And so this is a very, very close competitor, I would say. And when we are developing cell therapies, we have to be aware that there's a very simple other approach to do cell therapy in the patient, actually. And so just to sum up, I think we see how interesting this field is, that we have proof of concept, that we have cell therapies being actually used in the routine practice or starting to be used in the routine practice. Many issues around that, and I think manufacturing, as I've tried to show you, is really, for me, closely linked to the clinical setting. Tell me how long you take for your cells to be made, and I can tell you if it's suitable for my patient. So timing is really very, very critical. And I think it's a very exciting field to see more development in the future, and we have to work very closely together from the design of the cells until the application, and I really like this catapult holistic approach to get everything together. Thank you very much.
So just to remind again, the questions will be afterwards. And then now we are going to have a shorter break. And then we are going to back here in 30 minutes. So see you.